Picture this. You are at breakfast. You have a bowl of cereal, or maybe you are making something that involves milk in a less direct but equally committed way. A latte, a smoothie, a piece of toast with butter, none of which you have thought about very hard this morning because it is breakfast, and you are a person who functions at approximately 40% capacity before 10 a.m. And then someone who is either a person you love or a person you are about to have complicated feelings about says something while you're mid-pour. You know milk is basically just filtered blood, right? And then they leave. They just leave. They go to get ready for work or school or whatever they were doing, and they leave you there with your cereal, your milk, and a sentence that you cannot unhear and cannot fully evaluate without either knowing more biology or successfully suppressing the thought entirely. You attempt the second option. It doesn't work. So now you are here. And the thing is, the deeply uncomfortable thing that you probably already half knew when you clicked on this, the claim isn't entirely wrong. And the explanation for why it isn't entirely wrong is one of the more interesting pieces of mammalian biology that rarely comes up in conversations about dairy products or nutritional labels or the relative merits of whole versus skim. Whether milk is actually filtered blood is a question that deserves a real answer. Not a reassuring dismissal, not a sensationalized confirmation, but the actual biology of what milk is, where it comes from at the cellular and biochemical level, and what the relationship between the circulatory system and mammary secretion actually looks like when you examine it carefully. Start with what milk actually is at the biochemical level, because the conversation about its origins requires knowing what you're trying to account for. Milk is a complex biological fluid produced by mammary glands in female mammals for the nutritional support of offspring. It contains water, fat, protein, lactose, minerals including calcium and phosphorus, vitamins including vitamins A, D, B12, and riboflavin, and various bioactive compounds including antibodies, growth factors, hormones, and enzymes. The composition varies significantly between mammalian species. Human milk is substantially higher in sugar and lower in protein than cow milk. Whale milk is extraordinarily high in fat to support rapid growth in cold ocean conditions, and the milk of different species is calibrated to the developmental needs of their offspring. The primary proteins in cow's milk are caseins, which form the protein matrix that gives milk its characteristic opacity and white color, and which coagulate during cheese making, and whey proteins including beta-lactoglobulin, alpha-lactalbumin, lactoferrin, and immunoglobulins. The fats are primarily triglycerides packaged in milk fat globules surrounded by a membrane. Lactose, the milk sugar, is a disaccharide synthesized specifically in the mammary gland from glucose and galactose. None of these compounds is blood. Blood plasma contains albumin, fibrinogen, globulins, glucose, lipoproteins, hormones, electrolytes, and metabolic waste products in a saline solution. The overlap between blood composition and milk composition at the level of specific compounds is limited to some shared proteins, particularly immunoglobulins, which are present in both blood and milk, and some shared precursor molecules like glucose that are present in blood and then processed into lactose in the mammary gland. So, in the strictest sense, milk is not filtered blood. But the claim isn't coming from nowhere, and the biology of where it comes from is worth examining because the relationship between blood and milk production is more direct than most people realize. The mammary gland is one of the most metabolically active tissues in the mammalian body during lactation, and its relationship to the circulatory system is central to understanding where the claim about milk and blood originates. Mammary glands are modified sweat glands. This is not a metaphor or a casual comparison, it is the actual developmental and evolutionary origin of the tissue. The mammary gland shares developmental origins with the skin glands, and the structural organization of the lactating mammary gland, secretory alveoli surrounded by myoepithelial cells drained by a ductal system that empties at the nipple, is a modified version of the secretory gland architecture found in sweat and sebaceous glands. The secretory cells of the mammary alveoli take up precursor molecules from the blood, synthesize milk components inside the cell, and secrete them into the alveolar lumen, where they are collected and transported to the nipple. This process of taking up blood-derived molecules, transforming them, and secreting the products into a different fluid is the core of mammary secretion, and it is the basis of the blood relationship that the provocative breakfast claim is pointing at. The blood supply to the lactating mammary gland is extraordinary in volume. During peak lactation in a dairy cow, the udder receives approximately roughly 400 to 500 liters of blood for every liter of milk produced. This is not a printing error. For every liter of milk that flows out of the animal, 400 to 500 liters of blood have flowed through the mammary tissue to provide the raw materials for that liter's production. The mammary gland is one of the most highly vascularized tissues in the lactating mammal's body, and the blood flow required to sustain milk production is a substantial fraction of total cardiac output. The mammary secretory cells extract specific molecules from this blood supply: glucose, amino acids, acetate, fatty acids, calcium, phosphorus, vitamins, and either package them directly into milk or use them as precursors for the synthesis of milk-specific compounds. Lactose, for example, is synthesized inside the secretory cell from glucose and galactose, both of which come from blood. The milk fat globules are assembled inside the cell from fatty acids extracted from blood lipoproteins or synthesized de novo from blood-derived acetate. The cellular process is active synthesis, not passive filtration. Milk is not produced by passing blood through a membrane and collecting what comes through on the other side. It is produced by the biochemically sophisticated transformation of blood-derived precursors into milk-specific compounds through a series of enzymatic reactions that are unique to mammary secretory cells. The distinction between filtration and synthesis matters biologically, but the raw material source, blood, is accurate. The immunoglobulins present in colostrum and milk represent the most direct transfer of blood components into mammary secretions, and they are worth examining in detail because they illustrate the relationship between blood and milk in a different way from the metabolic precursor story. Colostrum, the first milk produced after birth, before the transition to mature milk, contains extraordinarily high concentrations of immunoglobulins, the antibody proteins that are also the primary immune defense circulating in the mother's blood. In cattle, colostral immunoglobulin concentrations can reach 50 to 100 grams per liter, compared to less than 1 gram per liter in mature milk. These immunoglobulins are not synthesized in the mammary gland. They are transferred directly from the blood into the colostrum through a receptor-mediated transcytosis process. The mammary epithelial cells, immunoglobulin molecules from blood vessels on one side of the cell, transport them through the cell, and release them into the milk on the other side. The immunoglobulins in colostrum are blood immunoglobulins, not precursors for blood immunoglobulins, not similar to blood immunoglobulins, but the actual blood-circulating antibodies that the mother's immune system has produced and that circulate in her blood as her primary defense against pathogens. In the neonatal period, these maternal antibodies provide the newborn with passive immune protection. The mother's immunological memory, accumulated over her lifetime of pathogen exposure and vaccination, is transferred to the offspring through colostrum to bridge the period before the offspring's own immune system matures. This is about as direct a blood-to-milk transfer as the biology produces. The immunoglobulins are blood components that become milk components through a cellular transport process. If you wanted to point to the component of milk that is most literally a blood component, this is it. Lactoferrin, an iron-binding protein present in both blood and milk, represents another category of shared components, though lactoferrin in milk is partially synthesized in mammary tissue rather than entirely transferred from blood. The overlap in blood and milk proteomes is real and extends beyond immunoglobulins to a range of bioactive proteins that reflect the mammary gland's integration with the circulatory system. The color of milk and the color of blood, and the obvious difference between them, is worth addressing because it is the most immediate visual argument against the filtered blood claim, and it deserves a real explanation rather than just being dismissed. Blood is red because of hemoglobin in red blood cells. Hemoglobin is an iron-containing protein that carries oxygen, and its iron porphyrin complex produces the characteristic red color. Milk is white because of an entirely different optical phenomenon. Milk is white because of light scattering by casein micelles and fat globules. Casein micelles, colloidal aggregates of casein proteins and calcium phosphate that range from 50 to 600 nanometers in diameter, are large enough to scatter visible light of all wavelengths approximately equally. When all wavelengths of visible light are scattered equally, the resulting color is white. The fat globules in whole milk contribute additional scattering. Skim milk with reduced fat content is slightly more transparent and appears less bright white than whole milk because fewer fat globules are present to contribute to the scattering. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. The white color of milk is therefore a physical optics phenomenon rather than a chemical composition phenomenon. It tells you about the particle size distribution in the fluid, not about the absence of blood-derived molecules. The red color of blood tells you about the presence of hemoglobin in red blood cells. Neither color claim tells you about the molecular relationship between the two fluids. Hemoglobin and red blood cells are not present in milk. The cellular filtering that occurs during milk secretion means that the blood's cellular components, including red blood cells, don't cross into the milk. The color difference is genuine and reflects real compositional difference. It does not refute the claim that milk's molecular precursors are blood-derived, because that claim operates at the molecular level of precursor chemistry rather than at the cellular level of which cells are present. The evolutionary origin of milk and the mammary gland provides context for the blood-milk relationship that the purely biochemical account doesn't fully capture. Mammary glands evolved in the early mammalian lineage from modified skin glands that secreted substances onto the skin surface, either as a form of skin moisturizing, as a microbial defense through antimicrobial secretion, or as a mechanism for transdermal uptake of secreted molecules by young who pressed against the parent's skin. The earliest mammary secretions were almost certainly not the nutritive milk of modern mammals, but rather antimicrobial or water-binding secretions that incidentally provided some nutrition. The transition from general skin gland secretion to specialized nutritive milk production required the evolution of the ability to synthesize lactose. Milk sugar is unusual in being found almost nowhere in biology except mammary secretion, and the progressive nutritional enrichment of the secretion. The evolutionary trajectory from incidental skin secretion to the highly nutritive milk of modern mammals took place over approximately 150 to 200 million years of mammalian evolution. Throughout this evolutionary trajectory, the mammary secretory cells maintained their dependence on the blood supply for raw materials. This is not a feature that was designed in, it is simply the consequence of the fact that all metabolically active tissues in a mammal depend on the circulatory system for their molecular inputs. There was never a stage of mammary evolution where the secretory cells were independent of the blood supply. The blood dependence of milk production is as ancient as the mammary gland itself. The question of whether milk is filtered blood therefore has to be evaluated against this evolutionary background. Milk is the secretory product of a tissue that takes blood-derived molecules as its primary raw material. It is not filtered blood in the sense that blood is passed through a membrane and collected on the other side. It is the metabolic product of a tissue that is supplied by blood in the way that all metabolically active tissues are supplied by blood. The specific molecules in blood that are most directly connected to milk production help illustrate the nature of the relationship in more concrete terms. Glucose is the primary energy substrate in blood, maintained by the liver and pancreas at relatively constant concentrations. The lactating mammary gland extracts enormous quantities of glucose from blood. In dairy cows, the mammary gland accounts for approximately 70 to 80% of total whole body glucose utilization during peak lactation. This glucose is used partly for cellular energy metabolism and partly as a precursor for lactose synthesis. Every molecule of lactose in milk began as glucose in blood. Amino acids circulating in blood, derived from dietary protein digestion and from protein turnover in other tissues, are taken up by mammary secretory cells and assembled into milk proteins according to the genetic programs expressed in those cells. The casein proteins that make milk opaque and that coagulate during cheesemaking are assembled in mammary cells from blood-derived amino acid precursors. The whey proteins are similarly assembled from blood amino acids, calcium and phosphorus, the minerals present in milk at concentrations that make dairy products significant dietary sources of these nutrients, are present in blood as ions and are actively transported into milk at concentrations substantially higher than blood concentrations. The mammary gland concentrates calcium from blood into milk. Milk calcium concentration in cow's milk is approximately 20 times higher than blood calcium concentration. The active transport systems that achieve this concentration represent a blood-to-milk mineral transfer that draws directly on blood mineral stores. During negative energy balance in lactation, when the animal is producing more milk than its dietary intake can support, the mammary gland draws on body reserves, bone calcium, adipose tissue fatty acids, muscle protein, all of which enter the blood and then flow to the mammary gland. In this scenario, the molecular components of milk are literally the former constituents of the cow's own body body tissues, mobilized into blood, and then directed to the mammary gland. The question of what colostral blood component transfer implies about the practical question of whether you're drinking blood when you drink milk deserves direct address because the provocative framing deserves a proportional response. The immunoglobulins transferred from blood to colostrum don't survive into commercially available milk. Pasteurization, the heat treatment that kills pathogens in commercial milk, denatures proteins including immunoglobulins. The three-dimensional structure that gives these proteins their immune function is disrupted by heat, rendering them biologically inactive. More practically, most commercially available milk is mature milk rather than colostrum. The colostrum produced in the days after birth is not what enters the commercial milk supply. Mature milk has substantially lower immunoglobulin content than colostrum, and the immunoglobulin component of mature milk, while present, is a small fraction of total milk protein. The direct blood-to-milk protein transfer that is most dramatic in colostrum is substantially reduced in the mature milk that constitutes the overwhelming majority of dairy consumption. The blood-derived precursors that constitute the molecular raw material for lactose, fat, and the major proteins, glucose, amino acids, fatty acids, have been transformed through multiple enzymatic steps into compounds that are chemically distinct from any component of blood. Lactose is not present in blood. The specific casein isoforms in milk are not present in blood. The milk-fat globule structure, with its specific phospholipid membrane composition, is not present in blood. The transformation from blood precursor to milk product is chemically substantial, so the claim that milk is filtered blood is wrong as a literal description of the manufacturing process but points at a real biological relationship. The more accurate description is that milk is the synthetic secretory product of a tissue supplied by blood, produced from blood-derived molecular raw materials through extensive biochemical transformation. The practical implications for how you think about milk, if you are someone whose breakfast was disrupted by this information, are probably minimal. Milk is what it has always been. A biological fluid evolved to support mammalian offspring development, produced through a biochemically sophisticated process by mammary glandular tissue, containing a complex mixture of nutritive and bioactive compounds that are demonstrably useful for the purpose they evolved for and for a range of nutritional applications in human diets. The blood connection is real in the sense that all the molecular raw materials for milk production are delivered by blood, and some blood components are directly transferred to milk, and the mammary gland's operation is absolutely dependent on a massive, continuous blood supply. The blood connection is not real in the sense that drinking milk is drinking blood. The compounds in blood and the compounds in milk are substantially different, and the transformative biochemistry of mammary secretion produces a fluid that is compositionally distinct from its source. If you find the relationship between blood and milk production disturbing at the level of personal dietary preference, that response is as valid as any other visceral food preference. Human food choices are not obligated to be exclusively rational, and the knowledge that something is biochemically transformed from another thing doesn't eliminate the effective response to learning about the transformation. But if you're looking for the factual answer to bring back to whoever said it at breakfast, the answer is kind of, in the most fundamental sense that all tissues depend on blood for their molecular inputs, and some specific blood components are directly transferred into milk, but the fluid you pour on cereal is not blood, and describing it as filtered blood is more provocative than accurate. The cereal is fine, the milk is fine. The person who said it is probably waiting to see if you bring it up again. You could. Or you could just pour your coffee and let the whole thing settle into the category of interesting biology that doesn't actually change anything about breakfast. Either choice is reasonable, the biology is the same regardless.